Mhc Class I molecules are a type of receptor expressed on all nucleated cells, which include B cells, T cells, macrophages, dendritic cells, and neutrophils. The intensity of the expression may vary from cell to cell. They are not, however, expressed on red blood cells. Mhc Class I molecules are composed of an alpha domain that spans the membrane. Together, the alpha 1 and alpha 2 domains form the wall of the groove where peptides bind. These peptides are only 8 to 10 amino acids in length. Mhc Class I is also associated non-covalently to beta-2 microglobulin, a soluble protein. Peptides bound to the groove are presented to CD8+ T cells. These molecules are synthesized in the ER and are properly folded into their quaternary structure thanks to calnexin. While it is a chaperone molecule, calnexin also protects Class I heavy chains from intracellular degradation, which improves the assembly efficiency.
Mhc Class II are found primarily on specialized antigen-presenting cells, such as dendritic cells and macrophages, as well as thymic epithelial cells. MHC2 mainly functions to present processed foreign antigen epitopes to CD4-positive lymphocytes. Activated T cells also express Mhc Class II. Mhc Class II is composed of 4 domains and 2 non-covalently associated alpha and beta chains, which both span the membrane. The alpha 1 and beta 1 domains form its peptide binding groove. This open groove is capable of binding a great variety of peptides anchored by the cleft, composed of polymorphic residues. There are 3 sets of Mhc Class II genes in the human HLA. This includes HLA-DP, alpha and beta, HLA-DQ, alpha and beta as well as HLA-DR, which is composed of alpha and 2 betas, which can form 2 different HLA-DR proteins.
Mhc Class I exists as a partially folded trimer in the ER and is bound by calnexin. Once beta microglobulin is able to bind to the trimer, we get dissociation of calnexin and reveal the partially unfolded Mhc Class I molecule. The Mhc Class I protein is then bound to chaperones calreticulin and ERP57. These chaperones are associated with the TAP protein complex, which is what allows for viral protein fragments degraded by the proteasome to enter the ER from the cytosol. If the Mhc Class I molecule binds to a peptide fragment, it is able to complete its folding and then is trafficked to the cell membrane for antigen presentation. Mhc Class II molecules are also synthesized in the ER, however they associate to form a complex with the invariant chain, and then this protein complex buds off into a vesicle. Vesicles are acidified into an endosome, which cleaves the invariant chain but leaves a clip protein in the binding groove of the Mhc Class II molecule to prevent binding to other proteins during transport. These endosomes are then able to fuse with other endosomes that contain degraded peptides. Before it can bind these peptides, the Mhc Class II molecule must associate with the HLA-DM protein complex, which removes CLIP from the peptide binding groove, allowing for MHC2 to bind the endocyte host peptides. Once bound to a peptide, the Mhc Class II molecule is then trafficked to the cell membrane for antigen presentation.
Class I and Class II MHC molecules are similar in 3D structure but have different subunit composition. While both are heterodimers, Class I consists of a membrane-spanning alpha chain non-covalently associated with a beta-2 microglobulin, and Class II consists of an alpha chain and a beta chain, both of which span the cellular membrane. The peptide binding cleft in Class I is made of the alpha 1 and alpha 2 domains, whereas in Class II, it is made of the alpha 1 and beta 1 domains. The Class II peptide binding cleft is also more open than in Class I and is able to bind to a greater variety of peptides. In the event of an infection, Class I MHC molecules are used to present bound peptides that are endogenous to host cells, while Class II MHC molecules present peptides that are exogenous in nature and are engulfed by the cell through endocytosis. The two classes also have distinct expression patterns. Class I molecules are expressed on all nucleated cells with a higher expression by hematopoietic cells. Class II molecules are expressed by a subset of hematopoietic cells and thymic epithelial cells. MHC molecules are an integral component of the immune system and are often the target of infection-causing pathogens. An example of such a pathogen is the human immunodeficiency virus, HIV. HIV is a retrovirus that infects host cells by inserting a copy of its own DNA into the host genome. Once the virus infects the host cell, its DNA encodes for accessory proteins that are then expressed within the host cell. These accessory proteins allow
which, once activated, are able to kill other cells that are infected with the virus that activate. MHC Class I is responsible for antigen presentation to CD8-positive T cells, which, if recognized, results in the CD8-positive T cells that activate and target cells infected with the virus. However, the blockage of MHC Class I allows for the virus to proliferate within cells without a response, and in this case, HIV proliferates within CD4-positive T cells, another key component to our adaptive immune response. As HIV proliferates within these T cells, it eventually lyses them to release more copies of itself, destroying the cell in the process. The destruction of the CD4-positive T cell population staggers the adaptive immune response completely, leading to the condition known as AIDS.